You're standing in the bathroom halfway through brushing your teeth and you catch your own face in the mirror. Maybe it's a small dimple that shows up when you smile, or that odd little valley in your chin, or the way your hairline dips down in a V at the centre of your forehead. A widow's peak, they called it in school. Features so familiar you don't even think about them anymore. They're just part of you, written into your face since birth. Except here's the strange part. Nobody really knows why you have them. Not your doctor, not your genetics textbook, not even the scientists who study human faces for a living. These tiny quirks, dimples and cleft chins and widow's peaks have been sitting in plain sight on billions of faces for thousands of generations, and we still can't explain where they come from or what they're doing there. If you're using this to fall asleep, that's exactly why I make these. So maybe subscribe and tell me where you are and what time it is. I read the comments and it's like Tokyo at sunrise, London at lunch, Los Angeles at midnight, all at once, each of you carrying your own face and your own story. Today, we're going to follow 3 of the most ordinary, most mysterious features humans can have. Cheek dimples, those little indentations that appear when some people smile, caused by a small anomaly in the muscle that pulls your mouth upward. Cleft chins, that vertical groove where the 2 halves of your jawbone didn't quite fuse perfectly before you were born. And widow's peaks, the V-shaped dip in your hairline where 2 invisible fields of hair growth suppression meet just a bit lower than usual on your forehead. You probably learned about these in school. Maybe you filled out a Punnett square. Dimples, dominant. Cleft chin, dominant. Widow's peak, dominant. Simple genetics. Neat little boxes. 1 gene, 2 versions. Done. But that story, the one they still teach in classrooms right now in 2025, is wrong. Or at least it's so oversimplified that it's barely true anymore. Because these traits don't follow the rules. They appear in children whose parents don't have them. They vanish and reappear as faces age. Some are there 1 year, gone the next. And despite a century of genetics, we still haven't found the genes responsible. So why do they exist? If evolution is supposed to optimize us, strip away the unnecessary, refine us into sleek survival machines, then why are we still walking around with little dents in our chins and forehead hairlines shaped like mourning hoods from the 1500s? The answer is stranger and more beautiful than any biology textbook let on. It's a story about how faces are built, one microscopic accident at a time, about how evolution sometimes just shrugs, and about how humans across centuries and continents have looked at these tiny, meaningless quirks and invented myths, omens, and entire systems of beauty around them. This is the story of the traits evolution forgot to remove. Or maybe the traits that were never problems in the first place. Picture the classroom. Maybe yours was beige walls and fluorescent lights. Maybe wooden desks that smelled like old varnish. The teacher's at the chalkboard or pulling up a PowerPoint slide, and there it is, the Punnett square. 4 neat boxes arranged in a grid. Along the top, capital D and lowercase d. Along the side, the same. Inside the boxes, combinations DD, 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 DD. And next to the square, the promise. Dimples are a dominant trait. If you have at least 1 D allele, you get dimples. 2 D alleles, no dimples. Simple as that. It felt good, didn't it? Like suddenly the chaos of human faces had rules. Like genetics was a puzzle you could solve with a pencil and some careful letter copying. The same lesson showed up for cleft chins. Capital C for the cleft, lowercase c for smooth. Widow's peak, capital W, straight hairline, lowercase w. The teacher might have asked you to look around the room, count who had which traits, calculate the percentages. You filled in your worksheet. You learned that inheritance was predictable. Tidy. Fair even. This wasn't some fringe teaching method. This is still happening right now in 2025 in high school biology classes and introductory college labs across the world. Educational materials, the kind published by major universities and textbook companies, list these exact traits as classic examples of Mendelian inheritance. Dimples, present or absent, dominant allele results in dimples. Cleft chin, C or C, one copy is enough. Widow's peak, W dominates over W. Students fill out Punnett squares, predict offspring ratios, answer quiz questions about what percentage of children will inherit a widow's peak if both parents are heterozygous. It's clean, it's satisfying, it teaches the fundamental logic of genes, and it's stuck with you. Decades later, you might still remember it. You might look at your own dimples or your child's smooth chin and think, "Ah yes, I know why that happened. One parent had the dominant allele, the other didn't." Mendel figured this out with pea plants. We figured it out with people. The human face decoded. Except here's the crack in the foundation. Sometimes children show up with dimples when neither parent has them. Sometimes both parents have prominent cleft chins and all their children have smooth ones. Sometimes a person's dimples are deep and obvious at age 7, barely visible at 20, and gone entirely by 40. Sometimes a widow's peak that looked sharp in childhood softens and retreats as the hairline matures, or appears out of nowhere in adolescence when it wasn't there before. The Punnett square doesn't predict any of this. A dominant trait isn't supposed to vanish. A recessive trait isn't supposed to appear from two dominant parents. And yet, it happens. Over and over. You might have noticed these mismatches yourself and assumed you'd miscounted, misremembered, missed some detail about your family tree. But you didn't miss anything. The model is incomplete. The authoritative genetics databases, the ones doctors and researchers actually use, now say something very different from that classroom chart. For dimples, there's no identified gene, no confirmed genetic mechanism, little rigorous research. Some geneticists now argue there's no solid proof dimples are inherited at all in any simple way. For widow's peaks, the scientific literature is blunt. Despite being commonly taught as a dominant trait, no studies actually support that claim. For cleft chins, family data collected over decades shows patterns that directly contradict the single gene story. Parents without clefts regularly produce children with them, far more often than a recessive model would allow. The Punnett square promised you answers. It gave you a framework, a way to think about inheritance that felt concrete and testable. And in that sense it did its job, but it also gave you a story that was too simple to be true. The real genetics of your face, the actual reasons you have or don't have a little dent in your chin or a V-shaped hairline, those turn out to be far messier and far more interesting. Because when the simple story falls apart, that's when the real questions start. If these traits aren't controlled by single genes, then what is controlling them? And if we've been teaching the wrong explanation for 100 years, what else about your face have we been getting wrong? So the simple story crumbles. The Punnett squares don't predict what actually happens in families. Children appear with traits their parents lack. Traits vanish between generations, then reappear. And when you go looking for the genes responsible, for the molecular mechanisms that textbooks confidently diagram with capital and lowercase letters, you find something unexpected. Silence, gaps, a humbling absence of answers. Start with dimples, those little dents in your cheeks that supposedly follow a clean dominant pattern. Here's what the genetics databases actually say. We don't know. We don't know which gene or genes create dimples. We don't know how they're inherited. There's been remarkably little rigorous scientific research on the question at all. Some older sources mention a possible location on chromosome 5, but that's speculation, not confirmation. No major genetic study has identified a dimple gene. No genome-wide association study has pinpointed variants that reliably predict dimples across populations. The trait that gets trotted out in every high school biology class as a textbook example of dominant inheritance. We can't even find it in the genome. This isn't a minor gap. This is one of the most commonly cited human traits in genetics education, used to teach millions of students the basic logic of Mendelian inheritance. and we have essentially no molecular evidence for how it works. Some geneticists now argue the entire premise is wrong, that dimples were mislabeled as a classic genetic trait decades ago, before we had the tools to actually check, and the label stuck because it was convenient for teaching. Not because it was true, but it gets more complicated. Because even when we can identify genes that influence facial features, the way they work doesn't match the Punnett square model. This is where modern genetics steps in with a vocabulary that sounds technical, but describes something you've probably already noticed in your own family: penetrance, expressivity, polygenic inheritance. Terms that sound like jargon but really just mean genes are messy. Penetrance is about whether a genetic variant shows up at all. In the old model, if you had the dominant allele, you got the trait 100% of the time. But real genes don't work that way. Many genetic variants have reduced penetrance. You can carry the variant and never develop the trait, or you can develop it only under certain conditions, or only later in life. This is why traits skip generations, not because of some hidden recessive magic, but because the gene that influences the trait simply didn't get expressed in one person, even though they carried it and passed it on. Expressivity is about how strongly the trait appears when it does show up. Some people have deep, obvious cleft chins. Others have just a faint dimple or a slight irregularity in the chin surface, barely noticeable unless you're looking for it. Same genetic background, totally different outcome. Variable expressivity means the gene's effect can range from dramatic to subtle to invisible. All in the same family, all from the same inheritance pattern. This is why your dimples might be pronounced at age 10 and almost gone by age 30. The gene didn't change, your face did. Fat distribution shifted, muscle attachments remodeled, skin thickened. The genetic influence is still there, but it's being filtered through decades of development and life experience. Then there's polygenic inheritance. Not one gene, but many, maybe dozens, each contributing a tiny effect. Most facial traits work this way. Not a single dominant allele that guarantees a cleft chin, but a collection of genetic variants that slightly increase the chance of incomplete fusion in the developing jaw, that subtly alter the growth rate of mandibular bone, that influence how connective tissue forms around the chin muscles. Add them all up and you might get a cleft. Or you might not. It depends on which combination you inherited, how they interact, and what else is happening during the specific weeks when your face is forming in the womb. This is where the old metaphor of genes as blueprints finally breaks down completely. Blueprints specify exact outcomes. If the blueprint says put a door here, you put a door here. Genes don't work that way. Genes are more like recipes. They provide ingredients. They suggest techniques, they set rough parameters, but the final dish depends on temperature, timing, the humidity in the room, how vigorously you stirred. Two people following the same recipe can produce wildly different results. For cleft chins, this explains everything the Punnett square couldn't. Why the trait appears in children whose parents don't have it, why it can show up on only one side of the chin, why it can be deep and dramatic in one sibling, barely there in another. It's not a single gene switching on or off. It's a developmental process influenced by multiple genetic factors, each with modest effects, all interacting with the contingencies of how your face actually grows. Widow's peaks show the same pattern. The textbooks still teach them as dominant. One copy of the allele, you get the peak. 2 copies of the recessive, you get a straight hairline. But when researchers actually looked for evidence, they found nothing. No family studies confirming the pattern, no genetic mapping identifying a widow's peak gene. One detailed review concluded bluntly, there is no published evidence that widow's peaks follow Mendelian inheritance at all. The trait exists, it's real, you can see it. But the genetic story we've been telling about it appears to be fiction. What's actually happening is probably something like this. During early development, your forehead hairline forms through a process involving multiple signaling molecules that tell hair follicles where to grow and where to stay dormant. The widow's peak is what happens when those signals create a V-shaped pattern instead of a straight line. Why they do that in some people and not others probably involves several genes affecting follicle placement, scalp growth patterns, and the timing of when certain developmental boundaries get established. And since hairlines continue changing throughout life, especially during puberty and aging, what looks like a widow's peak at age 12 might soften or sharpen by age 25, not because your genes changed, but because the development process kept going. This isn't a failure of science. This is science working exactly as it should, recognizing when old models don't fit, admitting uncertainty, building better frameworks. The reason we don't have a tidy list of dimple genes and cleft chin alleles isn't because researchers aren't looking, it's because human faces are constructed through fantastically complex developmental processes involving hundreds of genes, intricate timing, and cascades of molecular interactions that we're only beginning to map. These traits are real, they're heritable, they run in families, but they're not following the simple rules we invented to make inheritance feel predictable. And honestly, that's more interesting, because it means your face isn't the product of a few coin flips, a handful of dominant and recessive alleles randomly combining. Your face is the result of an unimaginably intricate process, a recipe written by evolution, cooked according to the specific conditions of your development, seasoned by chance. The dimple that appears when you smile, the little dent in your chin, the way your hairline traces a point on your forehead, these aren't genetic certainties. They're small accidents, tiny variations in a process so complex that even now, with all our sequencing technology and molecular tools, we're still trying to figure out how it works. To understand why these traits exist at all, you have to go back, way back, to around day 19 of embryonic development, when you were smaller than a grain of rice and your face didn't exist yet. Not as a face, anyway. What existed was a collection of tissue buds, little swellings of cells called facial prominences, growing out from the primitive head region like architectural elements waiting to be assembled. 5 of them initially. 1 frontonasal prominence in the middle, 2 maxillary prominences on either side, and 2 mandibular prominences below. All of them made mostly from neural crest cells, those remarkable wandering cells that migrate out from the developing nervous system and build much of your face. These prominences have one job: grow toward each other, meet with precision, and fuse. If they do it right, you get a normal face. If the timing is off by even a little, if one prominence grows slightly faster or slower than its partner, if the cells don't quite reach each other in time, you get variations, sometimes benign quirks, sometimes more significant differences. The face is essentially a construction project being executed by multiple teams working from different corners, all trying to meet in the middle on a very tight schedule. There's no foreman, no blueprint, just chemical signals diffusing through tissue, telling cells where to go and when to stop. The mandibular prominences, those 2 lower buds, are responsible for your jaw and chin. They start on either side of the primitive mouth and grow toward each other along the midline. By the 4th week of development, they should meet and fuse, creating a continuous lower jaw. Most of the time they do. The bone and soft tissue merge seamlessly. The chin forms as a single smooth structure. But sometimes they don't quite smooth out completely. The fusion happens, enough to create a functional jaw, but the midline remains slightly indented. A groove persists where the 2 halves came together. As the face continues growing, as the mandible elongates and the chin develops its adult shape, that little groove remains. It might deepen, it might stay subtle, but it's there, a signature of incomplete fusion visible decades later. That's your cleft chin. not a gene switching on, not a dominant allele, just two pieces of tissue that came close enough to work, but not close enough to erase the seam. The technical term for what didn't quite happen is fusion of the mandibular symphysis, the joint where the two halves of the mandible meet. In most people, the mental tubercles, those little bumps of bone on either side of the midline, grow together and blend. In people with cleft chins, they remain slightly separated or asymmetric, leaving a depression between them. It's a near miss in biological architecture. Close enough for a functional jaw, not quite close enough for a smooth chin. Now move up to the cheeks. The dimples that appear when some people smile are something else entirely. They're not about bone fusion, they're about muscle attachment. Specifically, a muscle called the zygomaticus major, one of the main muscles of facial expression. It runs from your cheekbone down to the corner of your mouth. When you smile, it contracts, pulling the mouth corner upward and outward. In most people, the muscle attaches to the skin as a single band, but in some people, during development, the muscle splits into two branches, a bifid zygomaticus major. One branch attaches normally, the other attaches more superficially directly to the dermis layer of the skin. When you smile and the muscle contracts, that extra attachment point acts like a tether. It pulls the skin inward, creating a dimple. It's a tiny variation in how muscle fibers connect to overlying tissue. Nothing to do with genes in the simple sense. Everything to do with the specific geometry of how your face assembled itself during those critical weeks. And because muscle attachments can shift over time, because fat distribution in the face changes as you grow, dimples can come and go. A child with prominent dimples might lose them in adolescence as facial fat redistributes. An adult might develop them later as the face thins with age. The underlying anatomy, that bifid muscle, might still be there, but whether it creates a visible dimple depends on everything around it. The thickness of the subcutaneous fat, the elasticity of the skin, how often you smile, which affects muscle development, a genetic trait that isn't stable, a feature that responds to life. Then there's the hairline, the widow's peak. This one operates on a different principle entirely. During development, hair follicles don't just sprout randomly across your scalp. They form in response to signaling fields, zones where particular molecules tell follicles to grow or stay dormant. On the forehead, there are two areas, called periorbital fields, where hair growth is actively suppressed. These fields expand outward from above the eyes, creating a border where scalp hair stops and forehead skin begins. In most people, these two suppression fields meet in the middle, forming a relatively straight or gently curved hairline. But if the fields are slightly smaller or positioned a bit farther apart, they don't quite meet. Hair extends farther down in the midline, forming a V-shaped point, a widow's peak. It's not a structure that grows, it's a space that didn't get suppressed, a gap between two zones of inhibition. This is the same developmental mechanism that's disrupted more dramatically in certain syndromes. Frontonasal dysplasia, for instance, involves failures of midline cell migration around those same early days, 19 to 21. The result can include widely spaced eyes, midline facial clefts, and a pronounced V-shaped hairline. The widow's peak you see in otherwise typical faces sits on the same continuum, a milder version of the same developmental variation. Not a disorder, not a problem, just the visible echo of how signaling fields were positioned when your forehead formed. All three traits, cleft chins, dimples, widow's peaks, emerge from the same fundamental truth. Building a face is an act of biological improvisation. The instructions are there, written in genes, but they're instructions for processes, not outcomes. Grow here, fuse there, attach this muscle, suppress hair growth in these zones. Each step depends on precise timing, on cells arriving at the right place at the right moment, on chemical gradients diffusing through tissue at exactly the right concentrations. And because all of this is happening in a wet, warm, slightly chaotic embryonic environment, where temperatures fluctuate and maternal hormones surge and cells jostle for space, the results are never exactly identical. Even in identical twins, even with the same genes. Think of it like a cathedral being built by multiple teams starting from different corners. They're all working from the same general plan, but small differences in timing, in the materials they use, in how they interpret instructions, leave quirks. An arch that's slightly pointed instead of perfectly round. A seam in the stonework that wasn't quite smoothed away. These aren't mistakes, exactly. The cathedral still stands. It's still beautiful. But it carries the signatures of its construction, the small accidents that happened along the way. Your face is the same. A structure built by processes that were precise enough to work, but loose enough to vary. And those variations, those tiny accidents of development, are what make your face yours. So let's stay with dimples for a while, because they turn out to be one of the best examples of how slippery the idea of a genetic trait really is. On the surface, a cheek dimple looks simple, a little hollow in the skin that appears when you smile, maybe clinging there faintly when your face is at rest. Something you can point to in a mirror and say, "I have it," or "I don't." The kind of thing the high school Punnett Square story loves. Big D for dimples, little d for no dimples, cheerful diagrams of dominant inheritance marching across generations. But if you go underneath the skin, the story stops being binary almost immediately. Under your cheek, between the skin and the bones of your face, lies a layered landscape of tissue, skin on top. Then a thin sheet of connective tissue called the superficial fascia. Under that, islands of fat, ligaments, blood vessels, nerves. Threaded through all of this are the muscles of facial expression, including the zygomaticus major, that long strip of muscle running from the cheekbone down to the corner of your mouth. Most of the time, the zygomaticus major is a single band. It starts near the zygomatic bone, dives through the fat and fascia and anchors near the mouth. When you smile, it tightens and pulls the corner of the mouth up and out. A smooth movement, transmitted through layers of soft tissue. In people with classic cheek dimples, that muscle often isn't a single band. It forks. Instead of one continuous strip, you can get a bifid zygomaticus major, splitting into two bundles as it travels toward the mouth. One bundle behaves normally, heading to its usual attachment. The other heads upward, more superficial, and grabs onto the underside of the skin itself. That extra attachment is the key. It acts like a guy wire anchored just beneath the surface. When you smile and the muscle contracts, that superficial branch tightens and pulls the skin inward at a very specific point. The surrounding fat and tissue get compressed. A tiny hollow opens up. That's the dimple. But notice what's doing the work here. It isn't a thing that grows like a miniature crater carved into bone. It's a relationship. Muscle to skin, muscle to fat, skin to the underlying fascia. Change any of those relationships slightly, and the dimple's shape, depth, or even existence can change. This is why dimples are surprisingly unstable across a lifetime. Look at babies. Many infants seem to have dimples, or at least pronounced cheek creases, especially when they smile or suck. But a baby's cheeks are dominated by something adults mostly forget they ever had: big buccal fat pads. These pads fill out the cheeks, help with suckling, and give that rounded cherub face people find so endearing. They also push against the overlying skin and muscles, exaggerating every little fold. If there's even a slight tether between skin and muscle, or a subtle line where the facial muscles gather, the buccal fat pad amplifies it. The smile digs in. A crease deepens. Parents look down, see dimples, and start counting them in the family trait inventory. Then the child grows. The buccal fat gradually thins as the face lengthens, the jaw grows, and the cheekbones become more prominent. The same tiny tether that created a dramatic infant dimple may now sit over a slimmer pad of fat. The tension redistributes. The dimple softens or shifts or disappears unless the smile is very broad. In some people, it vanishes so completely that it becomes a story. You had the cutest dimples when you were little. The reverse can also happen. Someone with no obvious childhood dimples grows into adolescence or young adulthood. Their face leans out, skin tightens differently over the cheeks. The muscles of expression bulk up with use. The bifid zygomaticus that never did quite attach like everyone else's finally has the right geometry. One day, in a photograph, they notice a small hollow that wasn't there before. Population studies suggest this isn't just anecdote. In one long-term study of Greek children, visible cheek dimples were found in a minority of kids, but the proportion appeared higher in adults. That's the opposite of what a simple, on at birth, off forever gene would predict. It looks instead like a feature that can be coaxed into view or eased back into the background as the architecture of the face changes. Weight shifts do the same thing. Gain fat in the cheeks, and any indentation has to fight its way through a thicker cushion. Lose fat and the underlying tethers become more visible. Aging adds another layer. Collagen in the skin breaks down, ligaments relax or tighten at different rates. Gravity pulls soft tissue downward. A dimple can migrate slightly, stretch into more of a vertical crease, or appear to deepen simply because everything around it has settled. The muscle variation may be the same from childhood to old age, but the landscape it pulls against is constantly being resculpted. Genes set the stage. The play runs differently every decade. Then there's the matter of where dimples appear. Some people have them on both cheeks, others only on one. Unilateral dimples, a quirk within a quirk. These asymmetrical dimples are a quiet reminder that left and right sides of the body are never truly mirror images. Development looks symmetrical from the outside, but underneath, cells on one side may divide a bit faster or migrate a little farther. Blood flow can differ slightly. Nerves can branch with subtly different paths. When the zygomaticus major is forming, it doesn't check in with its counterpart on the other side and ask, are we doing this bifid thing or not? Each side builds itself according to local conditions and genetic instructions interpreted in real time. One side might split, the other might not, or both might split, but the overlying fat and fascia might only allow one of those splits to create a visible dimple. Studies that count cheek dimples in adults find roughly half of people with dimples have them on both sides, and half on just one. Some samples show a slight bias to the right, others earlier reported more on the left. The numbers shift by population and sample size. What doesn't shift is the bigger lesson. Asymmetry is normal. A face that isn't perfectly balanced left to right isn't broken. It's simply honest about how development actually works. And then, not content to wait for developmental accidents, humans did something very human. We learned how to manufacture dimples. Modern cosmetic surgeons perform a procedure called dimpleplasty, a kind of deliberate imitation of that bifid muscle tether. The operation is usually done under local anesthesia. A small incision is made inside the mouth, in the lining of the cheek, no cuts on the outside. The surgeon removes or cores out a tiny circle of tissue from the inner cheek muscle and mucosa, then passes a suture through, tying the inner tissue to the underside of the skin at the spot where the new dimple is desired. At first, as the stitch pulls tight, there's a visible indentation even at rest. It can look odd, too deep, too fixed, as if the face is permanently half smiling. Over a few weeks, as the tissues heal and the scar between skin and muscle matures, the dimple softens. In many patients, it becomes dynamic, showing up mostly when they smile, fading at rest. exactly like the natural tethers it's copying. In surgical reports, the results are often described as long-lasting, even effectively permanent, but even artificial dimples live in the same changing landscape as natural ones. They sit in cheeks where fat gain, fat loss, aging and gravity keep rewriting the contours over decades. A procedure that cost thousands of dollars and took half an hour in a clinic becomes just another variable in the long story of a person's face. The existence of dimpleplasty says something revealing about us. We are willing to endure needles in the mouth, swelling, soreness, a few awkward weeks of too deep indentations, all to acquire a trait that biology usually assigns at random and then treats casually, letting it ebb and flow with growth and age. In some cultures and eras, dimples have been read as signs of youth, approachability, mischief, or luck. In others, they barely register. But the desire to create them on demand is a reminder that cute and charming can feel as compelling as strong or healthy when it comes to how we edit our faces. And underneath that desire lies one more quiet challenge to the classroom story. If you can be born without dimples, grow up to develop them as your cheeks change, or walk into a clinic and pay to have them sewn into place, what does it mean to call dimples a genetic trait? Genes matter. They influence how your muscles branch, how your fat is distributed, how elastic your skin is, how your face responds to decades of expression and gravity. But they don't write a single word on your cheeks and then walk away. They write a process, a way of building tissue that stays responsive to hormones, nutrition, habits, age, and even deliberate surgical intervention. Dimples are a moving target. They appear, fade, shift, deepen. They pick a side or choose both. They can be inherited, improvised, or purchased. They sit exactly at the boundary between what your DNA makes possible and what your life and your culture decide to do with that possibility. And they're not the only facial feature like that. Move your attention a little lower to the very bottom of the face, and you find another tiny variation that people have been speculating about for centuries. A groove where two halves of a bone once met. A small well that poets and casting directors have both fallen into. The human chin and the cleft that sometimes divides it. The first strange thing about that little well at the bottom of the face is not the cleft. It's the chin itself. If you lined up all our living primate cousins and asked them to show their profiles, you'd see something curious. Monkeys with snouts that slope forward, apes with strong jaws, but the lower face flowing back in a smooth curve toward the neck. Inside, some of them have a thick bony ledge called a simian shelf, a sort of internal buttress reinforcing the teeth and jaw. But from the outside, there's no jutting ledge of bone, no little platform at the front. Only humans have that. A true, forward-projecting chin, the mentum. A bit of bone that sticks out beyond the lower teeth like a balcony at the very edge of the skull. It's so familiar that it's easy not to notice how odd it is. We draw cartoon faces for children and absent-mindedly add a chin. We carve statues and assume the jawline will end with a neat little protrusion. Yet if you handed that skull to an alien biologist and asked them to look for distinctive traits, the chin would be one of the first things they'd circle in red. Even in our own family tree, it seems to be a late arrival. Neanderthals, for all their impressive strength and big brains, generally did not have a modern human-style chin. Their jawbones tended to sweep back more smoothly. Earlier members of the genus Homo show only tentative hints. The clear, strongly projecting mentum we take for granted becomes common only with anatomically modern Homo sapiens, within roughly the last couple of 100,000 years, though the exact timeline is debated. So this peculiar ledge of bone is new, rare in the animal kingdom and shared by every human alive today. You might expect that such a distinctive feature would have a clear, agreed-upon evolutionary explanation. It does not. For more than a century, anatomists, anthropologists, and evolutionary biologists have proposed theories, run models, and argued in journal articles about why we have chins at all. None of the explanations has won a decisive victory. One idea is that the chin is a kind of brace. As our ancestors began chewing different foods, maybe softer or prepared ones, or as our faces changed shape, the pattern of forces travelling through the jaw might have shifted. The chin in this story is reinforcement, a structural adaptation to resist stresses from chewing, talking, or even the bending forces that run along the jaw when we bite down hard. It's an appealingly mechanical story, a beam under load, a buttress where curves meet. But when researchers model those forces in detail, the results are ambiguous. Some simulations suggest the chin actually sits in a zone where tensile forces, pulling, not compressing, are strongest, which makes it a slightly odd place to put extra bone if you're trying to reinforce against classic crushing loads. Others argue the extra bone could still help redistribute strain. The data don't clearly point to the chin evolved as a chewing brace as a settled answer. Another suggestion is more social. Maybe the chin is a billboard. In this version, as human faces shrank and we began relying more on social cues, subtle expressions and long-term pair bonding, certain aspects of the lower face became signals in mate choice. A strong projecting chin might have been read, consciously or unconsciously, as a sign of robust development, hormonal balance, or just good genes. If enough potential partners preferred it, the trait could have spread through sexual selection, even if it did nothing particularly special for chewing. This story fits well with our sense that faces matter in attraction, and that jaws and chins in particular carry a lot of visual weight. But it runs into its own problems. When scientists look closely at human skulls from different parts of the world, what they see isn't one clear, ideal chin, but a riot of variation. That's where a PLOS One study comes in. One that did something deceptively simple. It measured chins. Researchers took skulls from multiple populations across the globe and quantified the shape and thickness of the mentum, the outline you'd see in profile, and the depth of bone at the midline. If there were a single universally attractive chin type being strongly favoured everywhere, you might expect those numbers to converge on a similar shape, even if faces differed in other ways. They didn't. Instead, chin shapes varied dramatically by region. Some groups averaged broader, squarer chins. Others tended toward narrower or more tapered ones. Thickness at the midline bounced around too. There was no obvious global best chin. No single contour quietly pulling all populations in its direction. The author suggested that this pattern is hard to reconcile with a story where a specific chin shape is under strong universal sexual selection. It fits more comfortably with a mix of gentler forces, genetic drift as populations move and split, local histories of who happen to have children with whom, and cultural preferences that may value different jawlines in different times and places. A third hypothesis for the chin steps back even further and says, maybe we're asking the wrong question. Over the last couple of million years, human faces have been retreating. Our jaws have generally become smaller. Our teeth have taken up less space. The mid-face has flattened. At the same time, the back of the skull has changed shape as brains expanded, and the base of the skull has flexed to accommodate an upright posture and a throat suited for complex speech. In the midst of all this architectural reshaping, the lower jaw had to keep housing teeth, anchoring muscles, and meeting the upper jaw correctly so the bite would still work. Some researchers now suggest that the chin isn't so much a feature that was added as a clever solution, but rather a leftover ledge that appears when a once larger face retracts. On this view, you don't evolve a chin because it's useful. You evolve a smaller, flatter face for a variety of reasons. Diet, breathing, brain case geometry, and the way the mandible remodels during development leaves a little bony shelf at the front. The chin is a side effect, a byproduct of many other changes, not an adaptation in its own right. These three stories, chewing brace, sexy billboard, developmental side effect, aren't mutually exclusive. The truth may be a weaving of all of them. A chin that arose because of how our faces shrank, constrained by the mechanics of bone and muscle, and then, once it was there, entered into the social realm as something people noticed, judged, and sometimes selected for. However it began, the modern human chin is not a single thing, it's a landscape. Within that landscape, some people have a smooth sweep of bone and skin, others have a noticeable groove or dimple running down the centre, the cleft. Anatomically, a cleft chin is usually a record of a meeting that never fully finished. During development, the two halves of the lower jaw grow toward each other at the midline and fuse into one bone. The overlying soft tissues do their own kind of handshake, knitting together muscles and skin. In many people, that fusion is seamless. In others, there's a slight indentation where the bone and soft tissue meet in the middle. Sometimes the underlying bone has a true notch. Sometimes the cleft is mostly in the overlying fat and muscle. Often it's a combination. The result can be a deep vertical groove, a small central dimple, or a faint suggestion that only appears in certain expressions. Family resemblance is strong. Cleft chins run in families often enough that they've been used as textbook examples of dominant traits. But, as with dimples, reality resists the neat boxes. Some children of two smooth-chinned parents show clefts. Some children of two clearly cleft-chinned parents do not. The pattern looks more like multiple genes and developmental variables nudging the jaw toward or away from a midline notch, rather than a single switch. If you overlay those individual stories on the global map of chin variation, what you get is a rich patchwork. In some populations, cleft chins are relatively common. In others, they're rare. The PLOS ONE data and other smaller surveys suggest that these differences line up at least as much with population history and drift as with any obvious survival advantage. In other words, a cleft chin looks a lot like a neutral structural variant that piggybacks on the uniquely human mentum. It arises from small differences in how the jaw halves meet and how the soft tissues drape over them, persists in some lineages, fades in others, and mostly escapes strong natural selection because it doesn't dramatically help or harm. If there were a powerful universal pressure to get rid of clefts, if they made chewing fail or breathing worse or survival clearly harder, you'd expect them to have been sanded away by now. Hundreds of thousands of years of evolution would have smoothed the midlines of our faces. Instead, the cleft lingers, unevenly scattered. A reminder that evolution is often indifferent to tiny quirks as long as the essentials keep working. That doesn't mean humans are indifferent. For all our talk about accepting ourselves, we're a species that edits its own face, and the chin is one of our favourite places to do it. Chin augmentation and reduction procedures, various forms of genioplasty, are among the more common facial surgeries worldwide. Some people feel their chins are too small, their lower faces receding. Surgeons slide or implant bone and synthetic material forward to create a stronger projection. Others feel their chins jut too far, or are too square or off-centre. Bone is shaved, repositioned, reshaped. Sometimes these operations are done for functional reasons, like correcting a severe bite problem. Often they are purely aesthetic, driven by internal ideals and external feedback about what a good chin looks like at a particular time and place. In some eras and cultures, a pronounced cleft has been praised, written about in poetry as a chin well, a tiny trap for kisses. In others, the same feature has been smoothed away in photographs or surgically softened. Designs change. The underlying bone changes only when we decide to take a scalpel to it. So in the space of a few centimetres of bone and soft tissue, you have something uniquely human in three different senses. A true bony chin that no other primate carries. A cleft that arises from small, mostly neutral developmental variations, and a canvas that modern humans continue to carve, fill and contour, as if evolution's long experiment with the mentum wasn't quite finished until we had our say. From here, if you travel upward from that ledge of bone toward the hairline, the story of little facial accidents doesn't stop. At the very top of the face, where skin, skull and scalp meet, another small irregularity sometimes appears. Not in bone this time, but in the border between hair and bare skin. A tiny V pointing down. Widow's peaks look like style choices. They resemble something a careful barber might have carved, or a costume designer's idea of how to make a character look intense. But the little V at the centre of the forehead is older than any fashion. It is not drawn in adolescence. It is traced before you have a face in the usual sense of the word. While the chin is being assembled from two growing halves of bone, something else is happening higher up. The skin that will become your scalp and forehead is deciding where hair will and will not grow. It doesn't do this one follicle at a time, improvising. It works in fields. Early in fetal life, the top of the head is a sheet of rapidly dividing cells. In that sheet, chemical signals begin to form waves and islands. Some signals say become hair, others say stay smooth. Systems with names like WNT, Sonic Hedgehog, BMP, and EDA/EDAR rise and fall in tiny gradients, like invisible weather fronts moving across a landscape that is still only a few millimeters wide. Where one combination of signals wins, a cluster of cells thickens into what developmental biologists call a placode, the starting point of a hair follicle. Where the opposing combination dominates, the skin remains bare. In most of the body, this tug-of-war produces patterns we barely think about. A scattering of vellus hairs on the forearm, denser growth on the scalp. But at the border between scalp and face, the pattern becomes unusually sharp. Above the eyes, along the upper forehead, the body draws a kind of ceasefire line. Signals that suppress hair formation spread out from the regions that will become the eyelids and eyebrows, creating bilateral no-hair zones that climb up the forehead. From higher on the skull, waves of yes hair signaling roll down. Somewhere in between they meet. The meeting point, where suppression just balances activation, becomes the outline of the hairline. In many people, those two hair-suppressing fields meet neatly at the midline, like two advancing fronts of high pressure finally touching. The result is a relatively straight or gently rounded border of hair across the forehead. In others, the picture is slightly different. The no-hair zones rising from each eye are narrower, or their upper edges meet a little lower, or a little farther apart. The central region where scalp signals dominate pushes farther downward before it gives way. The line between hair and bare skin is not smooth. It bows forward in the middle. That projected point is a widow's peak. Nothing dramatic has happened. No extra follicle has been specially summoned for the job. The follicles were always going to be there, because the scalp field that produces them is still intact. What has changed is where the advancing "don't grow here" territories call a truce. In other words, a widow's peak is a small administrative error at a border drawn in the dark. Once that border is set, it is stubborn. Unlike the teeth, which erupt in waves, or the bones of the face, which remodel throughout childhood, the basic population of hair follicles on your scalp is essentially fixed before you're born. Individual follicles cycle through phases of growth and rest. They thicken and thin. Under the influence of hormones, some miniaturize and eventually stop producing visible hair. But the skin of a typical forehead does not suddenly decide at age 30 or 50 to sprout a new outpost of scalp hair in territory that has been bare since birth. That's why, if you look back at childhood photographs, the outline of your hairline is remarkably consistent. Higher or lower depending on age and hairstyle perhaps, but recognizably yours. A widow's peak present in a toddler is usually the same little V that will appear in the mirror decades later, altered mostly by what has been lost around it, not by anything new being added. This also explains one of the small confusions around the trait, peaks that seem to appear out of nowhere. As people age, many experience recession of the hairline at the temples. Follicles on the sides thin or vanish under the influence of androgens. The central front for a while may be less affected. What was once a straight line across becomes an M shape. 2 bays of hair loss with a peninsula of hair still projecting in the middle. To a casual observer or to the person watching their reflection with some alarm, it can look as though a new widow's peak is emerging with age. In reality, no new peak has formed. The central portion of the original hairline has simply been left behind as the surrounding coastline erodes. The reverse can also happen. Someone born with a distinct widow's peak may lose so much frontal hair that the V is blunted or erased. What remains is a higher, more diffuse border. Again, the follicles didn't move. The pattern was there from the start. What changed was survival, which follicles held on and which gradually shut down. Genetics classes used to present widow's peaks as another neat example of simple inheritance. Big W means you have one, little B means you don't. Put parents on a Punnett square and let the ratios fall as they may. But as with dimples and cleft chins, the closer biologists look, the fuzzier that picture becomes. Families don't always follow the tidy dominant recessive script. Peaks can be faint, ambiguous, or only obvious if you pull the hair back a certain way. Large genetic studies naming the widow's peak gene have not materialized. Instead, what we see fits better with many genes pushing the signaling fields a fraction higher or lower, a millimeter this way or that, layered on top of the general architecture of the forehead. If that were the whole story, widow's peaks would be nothing more than minor cartographic quirks in the borderlands between hair and skin. But, as with the chin, there is a spectrum. At one end are the ordinary variations you see in a classroom or on a subway. Some people with straight hairlines, some with arcs, some with delicate V's. At the other end are conditions in which the shape of the hairline is one of many clues that early facial development took a different path. In frontonasal dysplasia, for example, the midline of the face doesn't come together in the typical way. Very early, when the front of the embryo is still more idea than face, cells that are supposed to migrate toward the center of the future forehead and nose can stall or veer off. Upward from them also start farther to the right, upward from them also start farther to the sides. The zone between them, where scalp hair is permitted, may extend lower. On the finished face, that can appear as a pronounced widow's peak, a deep central intrusion of hair into the forehead, sitting above a nose and eyes that are clearly unusual in other ways. Other syndromes tell similar stories in quieter accents. Donny-Barrow syndrome, Aarskog-Scott syndrome, Waardenburg variants, Opitz G/BBB. Each has its own genetic causes and constellation of findings, differences in spacing of the eyes, nose shape, sometimes hearing, sometimes development of the brain. In many of them, a distinctive midline hairline is part of the clinical description. The same chemical weather that drew an atypical outline for the skull and soft tissues also drew a slightly different border for the scalp. For the families living with these diagnoses, a widow's peak is not a charming quirk. It is one small piece of a much larger medical landscape. Yet scientifically, the connection is valuable. It reminds researchers that the way hair and skin divide the territory of the forehead is not an isolated cosmetic choice. It is tightly coupled to how the underlying bone, cartilage and brain case are laid out. By studying the extremes, the children whose hairlines curve in unusual ways, alongside differences in the midline of the face, geneticists can begin to map the signaling pathways that build the front of the human head. Which genes, when nudged, move the periorbital no hair zones? which alter the migration of neural crest cells, those restless builders of facial bone and cartilage. Which pathways link a change in one to a change in the other. Once you have those maps, even the ordinary variation starts to look different. The softly curved hairline you see in the mirror before bed is not just how your hair grows. It's the visible edge of a developmental compromise struck in the first weeks of your existence, when overlapping waves of molecules negotiated where scalp would stop and face would begin. Evolution, as far as we can tell, has very little opinion about whether that compromise leaves you with a straight line or a slight V. Widow's peaks don't seem to make chewing easier, breathing better, or survival in a drought more likely. They probably drift through populations much as cleft chins do, carried along by the genes that really matter for building a functioning head, allowed to persist because they don't, on their own, cause enough trouble to be weeded out. Culture fills the silence. It links that tiny notch of hair to mourning caps and superstitions, to villains and romantic leads, to stories about temperament and fate. Biology has its own quieter narrative. A widow's peak is a record of how the architecture of a forehead was negotiated, of how suppression fields met and almost met, of how the front of a skull and the edge of a scalp agreed on a boundary line that would stay with you for most of your life. Just as the little dimple in a cheek or the groove in a chin marks where tissues once reached for each other and stopped, the V in a hairline is the echo of an ancient, meticulous construction project. One more small reminder that the face in the mirror is not a single design, but a patchwork of tiny decisions made in the dark long before you opened your eyes. Evolution, in that sense, is a very selective storyteller. It cares deeply about some things. Whether lungs extract enough oxygen, whether bones are strong enough to carry a body, whether a brain can navigate a harsh landscape and a difficult social world. It is much less invested in whether a hairline curves in a V, or whether a muscle in the cheek takes a slightly crooked path. To see why, you have to shift from the close-up view of faces and families to the wider statistical view that population genetics takes. Instead of asking, "What does this trait mean?" you ask, "What actually happens to it generation after generation as people live, die, and have children?" From that distance, a lot of our favourite details vanish into the background. In evolutionary theory, there is a simple but powerful category: neutral traits. A genetic change is called neutral if, as far as we can tell, it doesn't make much difference to an organism's chances of surviving or leaving descendants. It isn't helping, it isn't hurting, it's just there. Neutral doesn't mean invisible. A neutral variant can give you a freckle in a particular spot, a slightly straighter thumb, a softer curve to the chin. It may be very obvious in the mirror and completely irrelevant to whether your ancestors got enough calories, avoided infections, or found willing partners. When a trait is neutral, its fate is not decided by how useful or beautiful it is. It is decided by chance. In each generation, not everyone has the same number of children. Not every child survives to adulthood. Some lineages simply get lucky and become common. Others dwindle and disappear. When the trait in question is neutral, it gets swept along in this random rise and fall. This process has a deceptively gentle name: genetic drift. You can think of it like this. Picture a slow, meandering river carrying thousands of small stones in its current. Some sit in calm backwaters and hardly move. Some tumble along the bottom for miles, being slowly rounded. Others are rolled up onto a sandbar and left there as the river shifts course. None of this is because the river likes one pebble more than another. The stones end up where they do because of eddies, little changes in flow, random collisions. Their shapes matter only if they are so large or so light that the water can't carry them at all. Everything in the middle, the ordinary stones, live out their histories under the spell of chance. Evolution has a similar feel. Big changes that break something crucial rarely last. organisms carrying them are quickly removed from the story. Big changes that help: better heat tolerance, more efficient digestion, sharper night vision, can, under the right conditions, spread astonishingly fast. But small quirks that neither help nor harm, those get tossed into the river and left adrift. Dimples, chin grooves, the exact outline of a hairline. Most researchers suspect these lie very close to that neutral zone, not because anyone has measured their precise selection coefficient. In practice, that would be extremely hard to do, but because nothing about them seems tightly linked to survival or fertility in any environment we know. They are decorative redundancies on a machine that was built for other tasks. Drift becomes especially powerful in small populations. Imagine the ancestral groups we visited earlier in the story. Bands of 20 or 30 individuals scattered across a difficult landscape, trading and intermarrying occasionally, but mostly surviving in local pockets. In a group that small, one bad year can remove half the adults. One successful hunter can father many of the next generation's children. If that hunter happens to have a cleft chin, there may be more cleft chins in the next cohort. Not because the cleft helped him hunt, but because he had other, more important advantages or simply better luck. Over thousands of years, these random bumps in representation can send neutral or near neutral